Okay, now that we have talked about DNA in more detail, we can now talk about some of the things that you can do with DNA, how we can study it, and what we can use or learn from studying DNA sequences. So, one of the most common ways to study DNA sequences is through cloning. Cloning, the word cloning really just means to make a copy of something, and you may have heard of cloning in the context of what we call organismal cloning, which really just means a clone in that context means an individual that's a genetic copy of some other individual. In fact, that's how most organisms that reproduce asexually, their offspring are technically clones of their parents. And then when we produce a clone of an organism in the lab, that means that usually a cell from an adult is used to make another individual that's genetically identical to the starting organism. But in this case, we're talking about DNA cloning, which means we're just making a copy of a particular DNA sequence, not the entire genome, not the entire organism, just part of one chromosome or one gene or whatever DNA segment we're interested in, and growing it or making copies of it in usually some kind of host organism, which can be perhaps bacteria or some other single-celled organism, usually another organism or a different species than the source organism, just a host that's easier to grow. So the first step in cloning DNA is getting your DNA of interest out from whatever context it's in. So that means, and there's different ways to do that depending on what your target DNA is, or your DNA of interest. So, for example, you can just directly synthesize DNA one base at a time. There are ways to do that, and of course that's the most difficult and labor-intensive method, because you're assembling each nucleotide. There are automated ways to make synthetic DNA, but the easier way is with PCR. So if you have a DNA, a small quantity of a DNA sequence that you want to clone, you can use PCR to first make just standalone copies of that sequence, and concentrate it, and then that makes it easier to do the rest of the cloning steps. Or once you have either your PCR generated DNA, or even your directly synthesized DNA, or even if you have DNA that's part of a naturally occurring chromosome, you can isolate it from the DNA that you're not interested in by cutting it out of that larger segment using special enzymes called restriction endonucleases. So an endonuclease is an enzyme that specifically cuts DNA. So it usually makes double-stranded cuts, so it cuts both strands, and by cut I mean it basically hydrolyzes the sugar phosphate backbone of both strands. And they specifically, or they look for specific DNA sequences when they cut. So we call them endonucleases because nuclease means they cut DNA, or they cut nucleotide backbones. Endo means they cut inside a strand, as opposed to cutting off segments from the end of a molecule. They cut somewhere in the middle. The reason they're called restriction endonucleases is because these enzymes mostly occur in bacteria, and their function seems to be mostly to cut the DNA of bacteriophage viruses. So bacteriophages are viruses that infect bacteria, and viruses, as you probably know, infect their host by injecting their DNA or sometimes RNA into the host, and then the host makes new copies of that DNA and new copies of the virus. But then these bacteria have evolved these endonucleases to cut the viral DNA and prevent it from replicating. So it's sort of like a part of the bacterial immune system. And we call them restriction endonucleases because the way that virus or bacteriophages anyway are studied is you grow a plate of bacteria and then you put the virus, the bacteriophage, on top of the bacterial host and let the virus replicate. And so when these bacteria though express these enzymes, these endonucleases, the enzyme restricts the growth of the virus. So that's why they're called restriction endonucleases. Sometimes they're just called restriction enzymes. But the reason that they are useful for cloning, again, is that they don't just cut randomly, they cut at specific sequences of DNA called restriction sites. So these are six. Every enzyme has its own site that it detects or binds to, and that's where it makes its cut. So over here, for example, this is an example of a sequence that has two restriction sites. One for this enzyme here called EcoR1, and mostly these enzymes are named after the bacteria they're isolated from. So Eco is short for E. coli, and R1 means restriction enzyme 1. That's just what the name means. And then PVU2 is from presumably some bacterial species that's abbreviated PVU. But each one has its own cleavage site or recognition sequence. So EcoR1, for example, recognizes this sequence, GAA, TTC. And meanwhile, PVU2 recognizes this sequence, CAG, CTG. And so when this enzyme detects this sequence of DNA in any DNA sequence, it will make a cut here at this point and here on each strand. And that creates what we call a sticky end, and it's called sticky because it leaves two single-stranded overhangs. So this sequence, AATT, now is a single-stranded set of 4 nucleotides that's sticking out over the end, and that happens on both strands. Meanwhile, PVU2 creates what are called blunt ends because the cleavage site is in the same position on both strands. Now, most of these, so these are just some more examples. Again, these are all different restriction enzymes, and most of them have what we call palindromic recognition sequences. So that means that they recognize DNA sequences that are palindromes. So, and remember we said that a palindrome, when we're talking about DNA, is a sequence that reads the same on one strand, going 5' to 3' as it does on the other. So again, for EcoR1, we just said the sequence is GAATTC, and that is the same exact sequence if you read it on the other strand, starting at the 5' end. So this would be the 5' end of the other strand. And so reading it from that end, we say it's GAATTC on that end too. And so all of these you can see are recognized palindromic sequences. But again, not just any palindromic sequence, just this particular sequence. And again, some of them have sticky ends because they leave single-stranded overhangs. Some leave blunt ends. And then the nice thing about these sequences too, especially the sticky ends, is that they can be they recognize each other, and then they can be rejoined by another enzyme called a DNA ligase, and we'll talk about that more in a minute. These restriction sites, again, they occur randomly. So if you look through any given chromosome, you'll find these sequences just distributed more or less all over the place. So in fact, you can say for any given sequence, so let's say for this enzyme BAMH1, it has a recognition site or cleavage site that's 3 or 6 bases long, and so you can say, well, if I have a chromosome that's X number of bases long, you can calculate on average how many BAMH cut sites there should be in that sequence. And but then there are some situations where you might have a nucleic acid sequence that you're interested in, and there is no restriction site, or at least the restriction site that you want is not present. So you can introduce restriction sites at the ends of a given DNA sequence by using PCR. So we talked all about PCR in the last chapter, and this is basically the same exact idea, except this time you're using primers that contain a restriction site. So this will be your primer. The orange part is the primer, but attached to the primer you have this sequence which corresponds to a restriction site. So in this case, it would be the EcoR1 cut site. So this sequence, this is the EcoR1 recognition sequence, and so your 5' primer has this sequence on it attached to the primer, and then the other primer has the exact same sequence but just on the other end. And so then when you replicate this sequence, the copies then will all contain these EcoR1 cut sites at both ends. And then you can use that later when you, after cutting this sequence with EcoR1, then you're left with these sticky ends. And so those then can be recognition sites for inserting this new this sequence that you just copied into a cloning vector. So even when your target DNA doesn't have naturally occurring restriction sites for the restriction enzyme you want to use, you can add them in. Now the next step, once you've gotten your DNA of interest, if you want to clone it, you need to insert it into a cloning vector. So a cloning vector is just another DNA sequence that has sequences that will allow it to be copied in the host organism that you're using to clone. So there are a lot of different vectors, a lot of different types of vectors. The most common type, most commonly used type of vector is a DNA sequence called a plasmid. So a plasmid is a small circular DNA molecule that occurs naturally in bacteria. So when I say small, we're talking about 5,000 base pairs or so. And these occur naturally in bacteria. So normally, you probably know bacteria have just one single circular chromosome, but that chromosome contains many, many genes and can be many millions of base pairs long. So these plasmids are much smaller than that, but they occur naturally in bacteria. Bacteria, in bacteria, they sometimes are involved in reproduction. So bacteria normally, of course, are asexual. They reproduce asexually, but sometimes some bacteria have the ability to sort of transfer DNA from one cell to another, and usually when they do that, they transfer these plasmids across. And so these plasmids, therefore, have some properties of a whole chromosome, but they're not nearly as big. So for one thing, plasmids have, by definition, a sequence in the DNA called an origin of replication. And all that means is that within this sequence, and this circle represents a plasmid. So it's basically a circular DNA molecule, and each little region here corresponds to a DNA sequence. So this is called the origin of replication. And this sequence is just a recognition sequence for the bacterial proteins involved in DNA replication. So we'll talk about DNA replication more later, but there's a whole bunch of proteins involved in copying a DNA molecule, and one of those one set of those is involved in starting the replication process. But those proteins have to bind to a specific DNA region within the DNA that sort of says this is where you need to start replicating from. So the proteins that replicate DNA won't just start anywhere. They have to start at these specific origin sites, or they're often just called ORI sites. And for the plasmid, the origin of replication is important because the plasmid needs to be able to replicate independently of the host chromosome. That's the whole point. So you're cloning, to clone your DNA, you want to make copies of it. And so this origin of replication is the sequence that will make sure that the host cell actually copies your cloned DNA. Then you also need some way to tell if the plasmid actually contains, or if your host organism actually contains your plasmid, and to tell if your plasmid contains your cloned DNA. So there are a number of different ways to do this, and we'll talk more about those later on, or in a few minutes. But for example, these antibiotic resistance genes are important because if let's say ampicillin is a antibiotic, and so and you probably know that an antibiotic is a drug that kills bacteria. However, many bacteria carry genes that give them resistance to those antibiotics. So an ampicillin resistance gene, and here it's just called AMPR, is a gene that codes for a protein. That protein would prevent the cell that carries the plasmid from dying when exposed to ampicillin. So you can, if you have a colony of bacteria and you want to know, and you're using this particular plasmid which has this ampicillin resistance gene, you want to know if this if your bacteria actually have the plasmid or not. You could apply ampicillin, and if they survive, that means they have the plasmid. In fact, the antibiotic resistance aspect of plasmids is interesting in its own right. You've probably heard of antibiotic resistant bacteria being a big problem in medicine these days. So there are more and more bacteria showing up, or people with infections by bacteria that carry antibiotic resistance with them. So normally, if you get a bacterial infection, you can go to the doctor. They'll give you a prescription for an antibiotic, and hopefully that kills the bacteria. But if you have an antibiotic resistant infection, then the antibiotic won't do anything. And these antibiotic resistant strains are becoming more and more common because as we take antibiotics, we increase the chance that just by chance you'll have a bacteria with resistance. And these plasmids play a role because these naturally occurring plasmids can also carry around these resistance genes. In fact, that's where these resistance genes come from. They've been isolated from naturally occurring bacteria that just by chance have these resistance genes. And you can have, and there are many cases where bacteria will pick up multiple different antibiotic resistance genes at the same time. So for example, this plasmid contains resistance to ampicillin and tetracycline. Tetracycline is another antibiotic. Again, this is an artificial plasmid, but there are naturally occurring plasmids that can have resistance genes for multiple antibiotics, and that's a growing problem too. Not just bacteria, because usually if you take an antibiotic for an infection and it doesn't work, then you can try a different antibiotic, and hopefully that will kill it. Because for most bacteria, there is more than one antibiotic that is effective. But if you have an infection with bacteria that contains multiple antibiotic resistance genes, it becomes harder and harder to treat that infection. And these plasmids are a problem because what happens is the bacteria can transfer plasmids from one cell to another. So for example, you could have one bacterial infection that carries ampicillin resistance, and then another bacterial infection at the same time that carries tetracycline resistance. And if the plasmid from one strain, the ampicillin resistant strain, transfers over to, say, the tetracycline resistant strain, now suddenly you went from having just one strain resistant to one antibiotic to having a strain that's resistant to two. And so these multiple antibiotic resistant strains are becoming more and more common and becoming a big problem, especially in hospitals, where people tend to pick up these kinds of infections, like after surgery, for example. But in any case, we use the antibiotic resistance genes just to select the plasmids we want. The last thing that the plasmid has to have is usually multiple restriction enzyme sites. And this is important because these are the places where you're going to cut the plasmid in order to insert your clone DNA. So again, you usually isolate your DNA and then cut it with a restriction site, a restriction enzyme, which will leave ends on it that are compatible with the restriction enzyme site of your plasmid. So If you cut your target DNA with, say, EcoR1, and then you cut your plasmid also with EcoR1, then you should get ends of those that are compatible with each other, and that allows you to target your clone DNA or your target DNA into a specific location within the plasmid. And so most plasmids have multiple cut sites, and some of them are actually inside these resistance genes, which will become, we'll explain why that would be later on. But those are important as well. And most of these are also just commercially available. So, for example, you don't have to make a plasmid or find a plasmid. You can go. There's any number of companies that will just sell these plasmids. So this one, PBR322, you can go to a lot of different companies and order this plasmid, and they just send it to you. The DNA dissolved in a solution in a tube, and then you can use it in your cloning. The nice thing about plasmids, again, is that they are small. So they're easy to work with. It's easy to get them into the host cell, and we'll talk a little bit about how that process works later on. The only downside is that because they are small, they're not effective for cloning large segments of DNA. So anything bigger than about 15,000 base pairs, it's difficult to use a plasmid to clone that. But 15,000 is 15,000 base pairs is big enough to deal with most genes and most significant DNA sequences. So, but this again just kind of illustrates how you would use a plasmid. So again, these are your plasmids, which have your antibiotic resistance genes and in this case, the PST1 restriction enzyme cut site. So you cut the plasmid with PST1, you've cut your target DNA with the same enzyme, PST1, and then you use this enzyme DNA ligase to seal them, connect them together. You transform your host cells, a strain of E. coli bacteria, and so the cells will now contain those plasmids. But now, and we'll learn that the transformation process is not 100%. So some of the cells will pick it up, some of it won't. Also, your ligation step will not be 100%. So when you mix your plasmid and your target DNA together, some of the plasmids will take up the target DNA, some won't. And so you'll end up with a mixture of cells, some that have the plasmid you want, some, so this plasmid here, some will have the plasmid without the insert, which would defeat the whole purpose, and then some will have no plasmid at all. And so those antibiotic resistance genes are where this is where the antibiotic resistance genes come in. So again, if this plasmid contains a tetracycline resistance gene, and that's the purple one here, then you can grow your cells on a colony or on a plate that contains the antibiotic tetracycline, and that will kill off all of the bacteria that did not carry the plasmid. But then you also want to separate out those cells that picked up the empty plasmid. So to do that, you use the fact that you've got ampicillin resistance. So we'll actually come back to how we do selection in a minute. First, there are other types of cloning vectors for making, especially for larger DNA segments. So a bacterial artificial chromosome is basically just like a big plasmid. So it's still a big circular DNA, still contains an origin of replication, contains a site or multiple sites that contain DNA restriction enzyme cut sites. So you can cut the plasmid and cut your target DNA. So the target DNA could be a piece of a chromosome, for example, like a eukaryotic chromosome, and incorporate it into the bacterial artificial chromosome, and then get those into the bacterial cells. The only really difference between the bacterial artificial chromosome and the plasmid is that in addition to the origin and replication, there are these PAR genes, which are basically just genes that are important for replication. So they can replicate on their own. They don't need the bacterial host genes to initiate replication. So they're just more easily able to replicate in a host cell. And then they also have these selection genes, so either antibiotic resistance genes, or in this case, they're using this gene called LAC-Z. Now, LAC-Z is not antibiotic resistance. Instead, these are genes that code for an enzyme that will let you visualize the cells. So basically, using this substrate called XGAL, any cells that carry that LAC-Z gene will turn blue, whereas those that don't will turn white. In this case, you put because you put your target DNA inside, your cut site was right inside that LAC-Z gene, the cells that carry this bacterial artificial chromosome will not express that enzyme, and therefore they will not turn blue when exposed to XGAL. And another type of artificial chromosome that you can use as a cloning vector is a yeast artificial chromosome. And the yeast artificial chromosome is similar to the bacterial artificial chromosome in that, again, you have an origin of replication, and you have multiple selectable markers, again, either some sort of resistance gene or some sort of visualization marker, and then multiple restriction sites. So multiple enzymes that can cut that restriction enzymes will recognize. The only other thing that a yeast artificial chromosome has are these telomeric segments, and a telomere is a part of a eukaryotic chromosome. So yeast are eukaryotes. They are single celled, but they are eukaryotes. They're actually a type of fungus, and so they are useful if you want to study large eukaryotic DNA segments or genes, because you can clone eukaryotic DNA into bacteria, but the bacterial cell will treat it like bacterial DNA. And so there are some features of eukaryotic genes that a bacterial cell or bacterial transcription system won't recognize, and we'll talk about what those are later on. So the yeast artificial chromosome is good if you want to look at those DNA elements that are only found in eukaryotes. So this new clone that you've made has these telomeric sections in it, and now your target DNA, these orange regions here. So this is the DNA you're trying to clone. And these telomeres basically just represent the ends of the chromosome. So that's an important difference between eukaryotic chromosomes and prokaryotic chromosomes, is that prokaryotes have circular chromosomes and eukaryotes have linear chromosomes. So by having those telomere segments, which are just DNA sequences that the host cell will recognize as telomeres, it's also there to make sure that the host cell will copy it. So then you transform those into yeast cells. The yeast cells that pick it up will start copying it, and then again, you've then cloned that DNA segment. And then one last kind of vector that's kind of for specialized experiments is an expression vector. So all of these, the vectors we've talked about so far, are really just to clone the DNA to make copies of it. But there are also cases where you might want your cloned DNA to not just be carried around by your host cell, but to actually be expressed. So if your cloned DNA is an actual gene that codes for a protein, you might want your host cell to actually make that protein, or to actually express whatever the gene is. So for that, you need an expression vector. The expression vector is basically the same as a cloning vector, except that in addition to your origin of replication and your selectable markers and your cut sites, sometimes the cut sites, restriction enzyme sites in a clone or in a plasmid or other kind of vector are grouped together in a region called a polylinker, basically just a stretch of bases that have a bunch of restriction sites back to back. So that means you can choose which enzyme you want to cut this plasmid and insert your gene. But on either side of that polylinker, there are the DNA elements you would need to actually express the gene. So like I said before, even if you're expressing or if you're cloning, let's say, a human gene in a bacterial host, the bacterial host, even though bacteria and humans use the same genetic code, the regions of the gene that the bacterial transcription proteins recognize are different than that of humans. So this plasmid contains these regions called the bacterial promoter and operator. And these are DNA sequences that the bacterial transcription machinery will recognize. So the proteins like RNA polymerase, which is the enzyme that makes RNA and actually starts the process of transcription, are recognize where to bind based on the location of these promoter and operator sequences. In addition, the expression vector would have genes that encode repressors that actually interact with these promoters too. So make sure that the vector is only expressed under certain conditions. But again, once you have, so you can use this vector to make the bacterial host not only carry around your clone DNA, but to actually transcribe it, meaning to actually turn it into RNA. But if you actually then want the host to take the next step and turn it into protein, your expression vector also has to have a bacterial ribosome binding site. So the ribosome does not bind to the DNA, of course, but once this sequence is transcribed, so once we have transcription, then the RNA, the messenger RNA, will of course contain your whatever you put inside this, whatever you inserted into your vector from your polylinker, but we'll also have a ribosome binding site. And so this is the location on the mRNA where the ribosome will know to bind. So your ribosome will bind here, and we'll start translating your protein along that mRNA. And then the last thing that your vector needs is a transcription termination sequence. So when the ribosome gets to the end of the mRNA, there has to be a sequence that specifically the bacterial ribosome will recognize as the termination region, where to stop. Not where the ribosome will. Not where the ribosome will stop, but the RNA polymerase, which makes the RNA, should stop. So, and again, we'll talk about how transcription and translation work in more detail later. But the point is that this kind of vector you would use if you want your host to not just carry the DNA, but to actually express the gene associated with that DNA. And again, you can use this no matter where your target DNA came from. So if your host is a bacterial cell, but your DNA came from a human or any other species, again, if you put it into this kind of vector, the bacterial cell will actually make a protein out of it, and it will be the same protein that would have been made in the original host DNA. So once you have your DNA of interest and then you have your cloning vector, you have to bring them together and make the recombinant vector. So the word recombinant just means you've combined DNA from two different places. So you've combined your host or your target DNA, which maybe came from a eukaryotic chromosome, and your cloning vector, which originated in a bacterial plasmid. And so the two of them together we call the recombinant vector. And so when we talk about recombinant DNA, that's all we mean. It's just a single DNA molecule that consists of sequences that originated in two or more different species or organisms. And attaching the DNA of the plasmid and the DNA, your DNA of interest together is done by an enzyme called DNA ligase. So DNA ligase, in a way, is kind of the opposite of a restriction enzyme. So DNA ligase looks for DNA molecules that have free ends, and it will look for either blunt or sticky ends. Although the sticky ends, if you cut your DNA with a sticky end, it has the advantage that they will recognize each other. So because if you cut your DNA of interest with EcoR1, for example, and you also cut your plasmid with EcoR1, the single-stranded overhang of the DNA of interest and the single-stranded overhang of the vector will match, and we'll be able to recognize each other. But there are other situations where you might want blunt ends, in which case it doesn't matter what enzyme either one was cut with. So you could in fact cut your plasmid with one restriction endonuclease and cut your DNA of interest with a different one, and if they both make blunt ends, then DNA ligase will just attach them together. In fact, that's one way to actually remove restriction enzymes or restriction sites from a plasmid or from a DNA of interest, is to cut both of them with different blunt end restriction enzymes, in which case the palindromic sequence would be gone. But in any case, whatever enzyme you use, DNA ligase will recognize the free ends and attach them together. So again, DNA ligase is a bacterial enzyme, and it's used by the bacteria to repair double-stranded breaks in DNA. So lots of things can make DNA molecules break or have double-stranded breaks, and enzymes like DNA ligase are there to repair those breaks. But again, we use them in cloning to attach our vector and our DNA of interest together. So then our combined recombinant vector now has a complete DNA circle. So and of course it reforms the sugar phosphate backbone, so it's actually creating a covalent bond between the plasmid DNA and your target DNA. And then the next step, once you have your recombinant vector, so that means your vector plus your DNA of interest, is to get it into a host cell. And this process is called transformation. So anytime you transform a host cell, you're just putting your recombinant DNA into it. And there are lots of different methods of transformation. This one here is just one of the more interesting ones. It's actually a way of sort of physically getting the DNA into the cell. It uses this device. It's actually called a gene gun. And essentially, you put your DNA that you want to transform onto these little tiny gold particles. Put that on top of a screen, and then you put the screen above your target cells. In this case, they're using plant cells, but you could do this with any kind of cells, hypothetically. And then above that, there's a little tube filled with helium gas, and then literally the device looks like a little gun, and you pull the trigger, and so a little puff of helium gas comes out, pushes, basically blows those DNA coated particles through the screen, and with enough force, some of the particles will actually puncture the cell wall of the cells and get into the cell. Now, of course, not all of them have to get through. Not all the cells have to be transformed. You only need, if you have a good vector, hypothetically, you only need one cell to actually pick up your vector and then start making copies of it. So this is just kind of a high-tech way of doing it. There are other methods. You can, especially for bacteria, you can just mix the cells and your vectors together, and bacteria will just pick up some of those DNA molecules spontaneously. There are ways to increase the efficiency of that. There's a process called electroporation, where if you just apply a weak electrical current to the bacteria, they form these big holes in the cell wall, and that will let the plasmid in. And there are lots of other methods. You can use viruses. So viruses are, of course, that infect bacteria. Do so by injecting DNA. You can artificially produce viruses that carry your recombinant plasmid and have the virus infect and transform your host cell. But however you do it, the goal is just to get the plasmid into the host cell so that the host cell starts copying your recombinant plasmid. But the last step I've already talked about a little bit is selection. So once you've transformed your cells, you have to select those cells that actually carry your plasmid. Because again, no transformation method is 100% efficient. So no matter how good it is, some of the cells will not actually pick up your plasmid. So you need some way to select the cells that picked it up. Again, with bacterial hosts, antibiotic resistance is an easy way to do selection. So you use a plasmid that carries one type of antibiotic resistance, and so in this case, I think they were using the blue region is tetracycline resistance. So if you grow your bacteria on a plate that contains the antibiotic tetracycline, then only those cells that express or that have the plasmid will survive. The rest will die. And so now you're left with a plate that contains just those cells that have the plasmid, or have at least the tetracycline resistance part of the plasmid. Now again, you could have cells that picked up a plasmid, though, that doesn't actually have the vector, because again, you combine your vector with your DNA to make the recombinant plasmids, but and you use DNA ligase, but DNA ligase doesn't know or can't tell the difference between the plasmid and the target DNA, unless again they have different ends. But even then, DNA ligase has the ability to combine them together. So DNA ligase is supposed to combine the recombinant or combine the plasmid with the DNA of interest to make your recombinant vector. But DNA ligase also will happily take the plasmid and just reconnect those ends back to each other that you cut with your restriction site. So it'll just reform the original plasmid. It can even do the same thing with your target DNA. So DNA ligase could take your target DNA and connect the ends of it to each other, and then you're left with this just sort of circular piece of DNA which contains your target DNA but nothing else, which makes it pretty much useless as far as cloning goes. And that can then get transformed into the host DNA, just like the recombinant plasmid did. So you need some way then to isolate those cells that picked up the plasmid but didn't get the recombinant plasmid. So they got basically the plasmid with no recombinant DNA in it. So to do that, you chose specifically, hopefully, a site to incorporate your target DNA that is inside one of those antibiotic resistance genes. So in this case, I think we've chosen a site that corresponds to, in this case, ampicillin resistance. So we chose a restriction site that's inside the plasmid, or inside the ampicillin resistance gene of our plasmid, which means that when we cut it with our enzyme, in this case PST1, the ampicillin resistance gene got cut, and then in the plasmids that picked up the foreign DNA, the DNA is now in between the two ends of that ampicillin resistance gene, which means it won't work. So if you take a gene and you cut it and then you insert a huge piece of DNA in the middle of it, that gene no longer functions because the bacterial cell will no longer produce a functional protein if the DNA segment has been separated like that, which means it won't produce whatever the protein is that protects the cell, in this case from ampicillin. So now you can grow your cells in a dish that contains ampicillin and tetracycline. Again, because you still want to kill off those cells that don't have any plasmid, but you can grow it in a dish that contains ampicillin. But of course, the downside of that is that it will kill the cells that you actually want. So the way that we grow or test for the presence of ampicillin resistance is the first thing you do is you grow your bacteria on a plate that is again covered or not covered in any antibiotic, but you grow them in colonies that are in precise positions. So there's different ways to grow bacteria. The easiest way is just to wipe the bacterial cells onto a dish. As the bacteria grow, as they replicate, they form basically just big piles. So bacteria don't move very much. So any given cell will just start replicating, and all of its offspring will just sort of be piled up around it. And eventually that pile will grow large enough that you can see it, and we call a visible collection of bacteria a colony. So when you look at your plate, you'll see a bunch of little circles, and each circle represents a colony of bacteria, and all of the bacteria in that colony are going to be the descendant of one individual cell, which means that they'll all be genetically identical. They'll all either carry your plasmid or not carry the plasmid within that colony. So, so if you want to select for ampicillin, or you want to select for the cells that are ampicillin sensitive, that means they will die in the presence of ampicillin, then you very carefully place the cells onto your dish in an array. So you might do a grid like this, and then you transfer those after you let those colonies grow up again with no antibiotic at all, and then you transfer those colonies to another plate. This is usually you do this with basically like a little stamp. So it's literally just like a piece of plastic or wood with some sterile cloth or paper on the bottom. You gently press that against your plate, and then you press, so some of the cells from each colony will get stuck to the stamp, and then you put that onto another plate, which has no bacteria, but does have your antibiotics like ampicillin, tetracycline, and then you let those grow up. And if there's ampicillin there, you look for places where there are no colonies. So like this little region here and this little region here, there's really much less growth. In fact, there's no visible colonies within those regions. So what that tells you is that if you go back to your original plate, which has tetracycline but not ampicillin, so go back and look at this plate at that same position, those cells will be the ones that or you can assume picked up the recombinant plasmid. So they're not, they will actually carry your cloned DNA. And then you use, a little Q-tip or pick, and you get some cells from that colony, put those in your incubation tube and grow them up and copy them, and voila, you now have cloned a DNA sequence. And there's this, again, this is how you do it with bacteria. There are other ways for selecting your host if you're dealing with yeast, or if you're dealing with plant cells, or whatever your host cell is. but you have to have some way of determining whether or not your host cell actually picked up your recombinant DNA. Okay, so we will stop there, and next time I'll talk about what you might actually want to do with your DNA once you have cloned it.